Hello, everybody. Thank you for continuing to tune in. Let's keep the ball rolling and discuss carbohydrates. Our learning goals will feature lots of identification. We will be identifying the many structures and many biochemical functions attributable to carbohydrates, and we will categorize how the many ways these sugars can isomerize ultimately influences their macromolecular diversity. And there will be a little bit of description on how these certain glycoconjugates can orchestrate cell signaling as well as recognition. Our molecule of the moment is simple if a little bit tedious. It's a group of molecules known as the hexoses. And these are really important to biology. They're the most abundant type of sugars, which is a form of carbohydrate. And they all have 6 carbons in them, hence why they're called hexose sugars or just hexoses for short. And we all know what glucose is. That's an example of a hexose, C6H12O6. You're going to quickly realize, if you don't know already though, that this molecular formula doesn't just spell out glucose. It spells out several of the isomers for the known hexoses. They have many, many isomers. And as a result, these are semi-complicated molecules, which makes them very tedious to draw. I want to bring to your attention something that's called Fischer projections. Maybe you talked about this in organic chemistry briefly, maybe you didn't, but you're probably familiar with the skeletal way of drawing an organic molecule, where you use lines, wedges, and dashes to sort of approximate the three-dimensionality of organic molecules and their functional groups. And as you can see, this is kind of cumbersome, and there's lots of stereocenters in carbohydrates, which can be a little annoying to draw repeatedly. So the scientist Emil Fischer came up with a way to sort of simplify this. What he did was say, okay, let's assume that we're going to twist the carbon-carbon bond angles to sort of get all these chiral functional groups on one side of the carbon-carbon backbone. All right, so it kind of looks to me like a centipede that was maybe pinned to a dissecting board. And so if we were to rotate this 90 degrees, it would then look like this representation, where we sort of pin that centipede down, belly up, and all of its legs are sticking out towards us. So we're going to convert all of these groups to wedges, and then the backbone becomes dashes. And since it is the same throughout all of these, what I'm going to call centipede leg groups, we're just going to assume that you know that any lines pointing in the horizontal direction are going to be wedges pointing up at you, and the vertical lines are going to be dashed pointing away from you. And this is a much, much faster way of drawing out really any carbohydrate, a hexose just being one of them. So why do I say all this preamble? Well, my challenge to you is this. Using glucose's Fischer projection as a starting point, I want you to find out how many dihexose isomers are possible. You can brute force this, or there's a way you can do a very simple mathematical formula to figure it out. There's many roads that lead to Rome, as they say. I want to emphasize, just to constrain the problem a little bit, pay attention to the dihexoses. Don't worry about the L for now. All right, and as a hint, I'm going to tell you that all D sugars are going to have the hydroxyl group on the carbon farthest from the carbonyl on the right. And you can see that the hydroxyl is indeed on the right side. So mull over that, and I will be picking on someone in class to share their thoughts. I think it's always nice to start with definitions, so let's refresh on what a carbohydrate is. It's actually a little hard to get a all-encompassing definition for it, but it's certainly one of those core classes of biomolecules that you just gotta know. And what I think is a decent definition for it is carbonyl compounds that also have two or more hydroxyls. And here's an important corollary, or compounds that would form carbonyl compounds with many hydroxyls upon hydrolysis. That stipulation is going to make sense once we take a look at more complex sugars like disaccharides or starches. Now, where does the word carbohydrate come from? It's semi-misleading. It came from early scientists who would do mass spec on carbohydrates, and they found that the ratio of carbon to hydrogen to oxygen in these compounds was 1 carbon, 2 hydrogens, and 1 oxygen. And they said, hey, I wonder if this is hydrated carbon, carbon that has water baked into some way, hence carbohydrate. Turns out it's more complicated than that. It's really more of a, I guess, coincidence that there seems to be a water molecule baked in. You know, it's not literally hydrated carbon. It's carbon that has a lot of hydroxyl groups. But the name stuck long after the myth was dispelled, as tends to be the case in science. So here we are today. But more importantly, carbohydrates are probably most known for being a reliable fuel source in biology. I feel like there was a time when carbohydrates certainly got demonized in the media and in a lot of popular science veins for being an evil compound that's bad for your body, but really that's not true. I'm going to say this a bunch throughout the metabolism lectures. Moderation is key, and you need carbohydrates. You just can't have oodles and noodles of it. But not only that, it's not just a fuel molecule. I mean, you know, I love sweet potatoes and any kind of potatoes, just like any other person. But carbohydrates also have structural roles. Okay? In that picture, that's a dung beetle. All right, they don't have an internal skeleton made of bones. They have an external skeleton, an exoskeleton, and it's not made of an inorganic material like calcium phosphate. It's actually made out of a carbohydrate, one that's kind of hard to digest. It's called chitin, and that provides not only a degree of structural support, but it allows water to condense on its external surfaces at certain times of the day, which it can then drink, which I think is a pretty neat non-structural application of a structural carbohydrate. But the more you know, right? The roles also extend beyond food and structure, signaling and recognition. That is such an important role of carbohydrates. The external environment of the cells contain lots of membrane-bound glycosylation patterns, and these really complicated branches serve as antennae that other cells can recognize as foreign or, you know, friendly. And sometimes not so nice cells learn how to disguise themselves with friendly looking carbohydrate chains and vice versa. So these are complicated molecules, and they are very important from the realms of nutrition all the way down to, you know, immunology, cancer biology, and etc. And I like to start with baby steps, talk about the simple versions of the biomolecule, and then expand. And the simplest kind of carbohydrate is a sugar. The fancy word for sugar is monosaccharide. And a monosaccharide is simply a carbohydrate that contains usually anywhere between 3 to 7 hydroxylated carbons. And of course, there's going to be a carbonyl usually thrown into the mix somewhere. We can subcategorize the monosaccharides into one of two groups depending upon where that carbonyl functional group is within the chain. If it is in the middle of the chain, and it can be, kind of on the fringe or dead set in the middle, doesn't matter. So long as we're dealing with a ketone, that means we're dealing with a ketose sugar. And fructose is an example of a very common ketose seen in biology. Generally, though, biology features a lot more aldoses, which is where the carbonyl group is situated at the end of the chain. And with Fisher projections, just by convention, we stick that carbonyl at the very top. And these are examples of some common aldose sugars. There's triose sugars, such as glyceraldehyde. This is the simplest carbohydrate, only has 3 carbons in it. Erythrose is a tetrose sugar. Ribose, I'm sure that sounds familiar, is a pentose sugar. And glucose, as we discovered, is a hexose sugar. And I know I said sugars contain 3 to 7 carbons, but I Googled it, and there are such things as octoses, so I don't think anyone is too strict with these definitions, but really they mostly fall within the 3 to 7 range. Hell, I can't even name a heptose sugar or 7-carbon sugar off the top of my head, which is probably an indicator of how prevalent and important they are in biology. You may have noticed that every sugar I've mentioned so far is the D-optical isomer. And like amino acids, that's not a coincidence. Sugars, more so than amino acids, are very chiral compounds. Take a look at these hexoses that have been drawn over here. And let's put the spotlight on glucose. How many chiral carbons does this molecule have? Well, there's 6 carbons present. 1, 2, 3, 4, 5, 6. But that top carbonyl carbon isn't chiral because it only has 3 different groups associated with it, and we need 4. And this bottommost carbon does have 4 groups to it, but there's 2 hydrogen groups which are the same, so that means there's an internal plane of symmetry. So that means carbons 2 through 5, the remaining 4, are going to be the chiral centers in that molecule. And if you think back to the amino acid lecture, well, amino acids, most of them only have 1 chiral center, which is the alpha carbon. So we got 4 times the amount of chirality in a hexose generally compared to something like an amino acid. So something I want you to be aware of is that, like I started off with, D carbohydrates are the preferred chiral isomer recognized by biology, and biology has trained itself to prefer L amino acids. And again, the word for this is homochirality. What's the benefit of making our molecules homochiral? Well, structural diversity is important, right? Because that lets you have many geometries and many functions. But too much structural diversity is chaos, and it can be hard to consistently get the structures you want designed. So by having carbohydrates also be homochiral, we reduce entropy. We cut down on the chaos a little bit and say, okay, of these chiral centers, we're really only going to toy with these ones. And you're about to see that you can get a lot of diversity by just flipping around the functional groups on those 3 carbons. Let's start with the group that has been highlighted in red. Let's invert the chirality of that carbon center. And to do that, we're going to take the hydroxyl on the right and swap it with the hydrogen on the left. And what happens when we do that? We're going to get a completely different sugar molecule. And the name for that is mannose. It looks just like D-glucose, but D-mannose is going to have the hydroxyl on the left. And now let's play this game again with the blue highlighted chiral center of glucose. And let's flip-flop those 2 functional groups and see what we get. Again, a completely different sugar again. We'd convert D-glucose into D-galactose. So by keeping carbon 5 homochiral consistently in the D configuration with the hydroxyl on the right, we certainly don't sacrifice a whole lot of diversity, which is a good thing. What I've just showed you is an example of the many epimers we can get out of sugar molecules. And you may remember from organic chemistry, or maybe you don't, that an epimer is a certain kind of diastereomer that only differs by 1 chiral center. So mannose is an epimer of glucose, and galactose is an epimer of glucose, but they are not epimers of each other. They would be diastereomers because they differ by more than 1 chiral center. Now, what if we were to do the unthinkable? Let's say we'll have our D-glucose molecule, and let's change all of the chiral centers, and we'll reflect them across this imaginary mirror and see what we get. By changing every single chiral center, including the forbidden one at the bottom, the one that biology normally doesn't touch, we are going to get a different molecule again, but it's not going to have a different name. Instead, it's going to have completely inverted chirality. We are going to go from D-glucose to L-glucose. D-glucose is recognized by biology. L-glucose is not recognized. This is the power of chirality. They're both glucose, but one has calories and the other doesn't. There's actually, there was a push at one point to try to make L-sugars, L-glucose, as a calorie-free sweetener like Splenda or Equal, but making L-glucose is really synthetically challenging and expensive, so that never really took off. So I think it's pretty interesting that you can futz with the carbons, you know, here, here, and there, and you can get biologically useful compounds, but the moment you touch that one, you run into problems from a biochemical sense. Now, the last thing I want to mention is glyceraldehyde. This one's kind of interesting, it being the smallest carbohydrate. It only has one chiral center. So its epimer is also its enantiomer. You don't get some other kind of sugar. You go from D-glyceraldehyde to L-glyceraldehyde. It's going to be a total diastereomer of itself because, again, it only has one chiral center. In addition to chiral diversity, the number of isomers possible, one sugar can also take on many different conformations. We've been sketching sugars like glucose in its linear form so far, like this. But monosaccharides in mild to basic solution pH will actually cyclize automatically. And what happens is kind of like the Ouroboros, that mythical serpent that's biting its own tail. The alcohol at one end can do a nucleophilic attack and swing around and form a bond with this electrophilic carbonyl carbon that I've highlighted there in red. So those backbone carbons can rearrange into this hexagonal shape to facilitate the following nucleophilic attack like so. And when the sugar does that, it's going to convert both of those functional groups into this composite one. And it's either going to be a hemiacetal or a hemiketal. And I understand it may have been a minute since you took organic chemistry, so we're just going to do a really quick crash course on that. When you combine alcohols with aldehydes, carbonyl is in a primary position, you will get a hemiacetal, which looks like that. The OH and the OR group attached to the same carbon. When the alcohol attacks a ketone, so this is going to occur in ketoses, and of course this will be aldoses. What happens in the case of a ketone and alcohol reacting? We get the hemiketal. The way I like to think of it as well is that hemiacetals, hemiketals, it's kind of like if an ether and an alcohol became roomies, kind of like, Bert and Ernie. So there's a couple of caveats to this cyclization reaction that I've just described. It is spontaneous, but it doesn't happen in all conditions and to all carbohydrates. For example, I said that it only occurs in C4 sugars or greater, tetroses, pentoses. Doesn't happen to trioses. And this cyclization reaction occurs in mild to basic solution pH. Does not work in acid. You will actually undo it if you add acid to a sugar. So what I want you to mull over is why. Why do you think that is? I'll let that haunt you for a little bit, and we'll touch upon that in class. Something that I want to haunt you with right now, though, is the directionality of this attack I've just drawn over here. All right. The electrophilic carbon in that reaction is sp2 hybridized, so it exists in a plane. And I'm going to do my best to draw in 3D here, like so. I'll draw out the oxygen all foreshortened. And I'll put that hydrogen in the back. And here are the backbone carbons, 4 more. The nucleophilic hydroxyl has options. It can swing up and around and attack the top of that trigonal carbonyl plane, or I'll try to do a different color here. It can swing around and attack the bottom. In either case, we are going from an sp2 hybridized system to an sp3 because the carbonyl ultimately gets converted into what? An alcohol moiety. So if that nucleophilic hydroxyl, which I'll make explicit there, attacks from the bottom, that is going to kick up the oxygen that will become the hydroxyl moiety. We see that over here. That ethereal oxygen used to be the hydroxyl. In the case where the hydroxyl swings up and attacks the top face of that electrophilic carbon, it's going to force the alcohol to point down, which we see over here. The OH points down, it's below the plane of the ring, and here, when we attack from the bottom, it knocks up the OH to above the plane of the ring. These are called anomers. And there are two variants, alpha and beta. It is said that the carbonyl carbon, or the C1 in the case of an aldose, is anomeric because it has the potential to become a chiral center. It's not chiral in the linear state, but when you cyclize, you turn it into a chiral center. Said more simply, an anomer is an epimer made via cyclization. For some reason, I have a very hard time remembering that alpha anomers has the hydroxyl pointing down below the plane of the ring, and beta, it's pointing up. So I had to come up with a trick that I'm happy to share with you. I want you to think beta balloons. And what do balloons do? They float up, assuming they're filled with a gas that's less dense than air, but you get the drift. Over the ring. So I hope you can begin to appreciate the structural messiness that carbohydrates bring to the table. I mean, first we started with isomers, and then the isomers can cyclize, so they can be linear or cyclic. And when they cyclize into the alpha anomer or the beta anomer. So there's many dimensions of structural complexity here. And what's more is that sugars can cyclize in more than one way. Most commonly, they can link up like the Ouroboros into 5-membered rings, which we call furanoses, or 6-membered rings, which we call pyranoses. And because that's not complicated enough, these structures are also in rapid equilibrium, or they mutorotate, because this cyclization reaction is reversible. So let's take a moment to examine the anomeric equilibrium observed for glucose. If we take glucose powder, let's say it's in a linear conformation, and we dissolve it in water, that's going to cause it to cyclize and mutorotate almost immediately. And once we reach equilibrium, about 1/3 is going to form the alpha glucopyranose configuration. I want you to pause the video and see if you can identify it in these 4 cyclic structures I've drawn on the right. What is the alpha glucopyranose? Right. Here is our hemiketal. I look for OCO, and it's pointing down. Beta balloon is pointing up. That's pointing down. So, alpha pyranose. Now, almost 2/3 of those glucose molecules partition into the beta glucopyranose configuration. Pause again and see if you can spot it. Again, I like to look for the anomeric carbon, which is the OCO, the hemiacetal or hemiketal, and I see it there. This one's pointing up. It's on the top side of that ring plane. It's a beta pyranose. And I know it's a pyranose because 1, 2, 3, 4, 5, 6. Don't forget to count the oxygen. It's part of the ring, even though it's not a carbon. It's a 6-membered ring. Now, if you're good at math, you may notice that 31 and 64 don't exactly add up to 100, and that's because 3% of the glucoses in solution partition to the linear state, and trace amounts, way less than 1%, adopt the furanose configurations shown over here. Measurable, but not really significant in a biological context. So the takeaway here is when you picture a glucose molecule in solution, it's not a static entity. It's interchanging throughout all these possible configurations. The next kind of carbohydrate we'll talk about are the disaccharides. These are sugar dimers joined via a special glycosidic bond. And a glycosidic bond essentially is a bond formed between the hemiacetal and or hemiketal of a sugar conjoined with something else. And I'm keeping that kind of vague intentionally. It can be another sugar, which would be a sugar-sugar glycoside, aka a disaccharide, or it can be a small molecule or a different biomolecule altogether. But we're going to just constrain the discussion for now to disaccharides. And I wanted to show you some 2 examples of very common disaccharides. This is a picture of lactose. It is a disaccharide formed between 1 glucose unit and 1 galactose unit, connected together with the glycosidic bond. All right. Lactose, you may be familiar with. It's milk sugar. It is abundant in dairy products. Another very common disaccharide is sucrose. This is a disaccharide formed by conjoining glucose again, but with fructose instead of galactose. And it's more apparent here that we're actually conjoining a pyranose sugar because this is a six-membered ring with a furanose, a five-membered ring. Fructose is a ketose and a furanose. And sucrose is more commonly known as table sugar, perhaps. This is the sugar used in your cookies and baking for general sweetening your tea and your coffee. A lot of people sort of just assume that that's glucose, and that's not true. It's sucrose, which is sweeter than glucose, but it does get digested into glucose and something else. And again, I'll take a moment to highlight the glycosidic bond here. Technically, it extends all the way up to this oxygen. And to that oxygen as well in that case. Now, you may notice that the way I highlighted those glycosidic bonds are a little bit different. And my trick for finding what used to be the hemiacetal or the hemicatal is I look for OCO. So there's an OCO over there. And this one's trickier. There's an OCO that kind of pops out easily in that regard. But there's another OCO like that in the case of sucrose. So that's pretty poignant. I want you to really study that because that brings us to the next topic within disaccharides, which is reducing versus non-reducing sugars. Let me ask you this question. Which of these do you think still has the capability of forming additional glycosidic bonds? Said another way, could we polymerize lactose? Can we polymerize sucrose? We can only polymerize one of them. And the answer is actually lactose. And that's because it has one additional hemiacetal over here, OCO. OCO. There is no free OCO, hemiacetal or hemicatal group in the sucrose molecule. That's because in the case of lactose, we formed our glycosidic bonds between the hemiacetal of glucose and a hydroxyl in galactose, which means that the hemiacetal of lactose is free and we can stick another group onto there, such as another sugar or another lactose unit. In the case of sucrose, it is the hemiacetal of glucose plus the hemicatal of fructose. And they are conjoined with each other, head to head, kind of. We have run out of hemiacetals and hemicatals in both sugar units because they are linked together. So we can't feasibly extend sucrose into a trisaccharide or so on and so forth, an oligosaccharide beyond that. When a sugar has a free unbonded hemiacetal or hemiketal, we call them reducing. Which means we can form additional glycosidic bonds on that unit. When they are not available to react, we call those non-reducing. And you may be thinking, well, why do we call it reducing and not something like reacting or reactable and non-reactable? And one of the reasons is we test for that with a redox reaction. It's called the Benedict's test. You've likely done this before. In general biology lab is a very common exercise where what you do is you subject the sugar to this basic Benedict's reagent, which contains blue copper 2 ions. The copper 2 ion is a mild oxidant, and the free hemiacetal can mutorotate partially back into an aldehyde, which is a mild reductant. So when you heat them, they're going to undergo a redox reaction where the copper is going to oxidize that aldehyde into a sugar acid, and in turn that's going to produce the red-colored copper wine. So that is why we call that a reducing end, because it has the capacity to reduce copper 2 to copper 1. Another property of sugars worth noting is their optical rotation. This naturally is a product of their chirality. Chiral compounds such as amino acids, sugars, etc. will rotate plane polarized light. And you can observe that phenomenon with a polarized filter and a polarized light source, like shown over here in this animation. Let's pretend that that mounted crystal in that clip is a sugar crystal. And notice how the polarized light that we're shining through it without that filter is, it passes through transparently. But when we rotate that filter at certain angles, it's going to deconstructively interfere and block out the transverse light wave. And that's why it appears black once you rotate it to certain degrees. And this is actually a really useful property because all sugars are going to rotate plane polarized light to different extents. And this is a really handy property if you want a way to identify a sugar without, I guess, tasting it. I mean, sugars for the most part are sweet, white powders, and lots of them are isomers of each other, so they all have identical mass spec signals. So discriminating sugars is quite a challenge, but this is a really convenient hack for doing that. Here are some examples. The sugar D-sucrose is going to rotate plane polarized light 66.5 degrees to the right, okay? And hence we use a positive sign to denote rightward rotation. D-glucose, which is a component of sucrose, rotates plane polarized light a little differently. It's still to the right, but not as extreme, positive 52.7 degrees. Now let's compare glucose to fructose, which is the other half of a sucrose molecule. That rotates plane polarized light completely differently. It's going to rotate it to the left and negative 92 degrees. This is kind of interesting. First of all, glucose and fructose, they're both C6H12O6 for starters. And all of these are D carbohydrates, but they very differently rotate this plane polarized light. Now, check this out. If you're into baking, bakers will often cook down sugar in acid, specifically sucrose, and intentionally break the glycosidic bonds holding together the glucose and fructose units. And it will also linearize the sugars in the process. This is going to produce something called invert syrup or invert sugar, which is basically just a gooey mixture of glucose and fructose. And it's sweeter than sucrose, and it's going to be less prone to crystallization. And that makes sense. It's a heterogeneous mixture of, two molecules, basically. Well, if it's a syrup, I suppose technically it's a homogeneous mixture, but those are two very important properties for candy making. If you like chocolate truffles, I know I do, they sweeten the interior chocolate ganache with inverted sugar, which helps keep that gooey ganache gooey because it's not going to crystallize and temper as easily. Now, the hard chocolate coating on the outside is going to be sweetened with table sugar, sucrose, because that is going to easily crystallize and make a firmer chocolate. And so you might be wondering, why do they call this compound, this mixture, invert syrup? Well, it has to do with optical rotation. Remember that sucrose optically rotates positive 66.5 degrees. Invert syrup inverts that sign. It's going to actually rotate negative 40 degrees. And why did we invert the way it optically rotates and to that degree? Well, if you sum up positive 53 and negative 92, you get around negative 40. Look at that. I think that's pretty neat. So the moral of the story is you can't like candy without liking biochemistry. I'm sorry, those are the facts. The next and largest breed of carbohydrate is the polysaccharide, which are polymers of these simple sugars. You can form these from the free reducing end of the sugar monomeric units, and by continuously clipping on additional units through glycosidic linkages, you can extend those chains to pretty dramatic degrees. I mean, some polysaccharides have molecular weights that exceed 20,000 Daltons in molecular weight. That's going to have at least 100 sugar units chained up together in a row. And we can further subcategorize the different kinds of polysaccharides based off of their shape and composition. Let's first consider the homopolysaccharides, which are the simplest. These are going to form when you take one sugar and iterate them within a polymeric chain. And you can do so in an unbranched fashion, just a single long line, or you can throw in a few branches or many branches. We'll talk more about these soon, but here's some nice illustrations that simply get that message across. And what are some general functions of homopolysaccharides? We'll get into the weeds soon enough, but off the top of my head, they're going to be really great energy storage compounds. If you want to have a glucose bank, let's tie them up together into one molecule so they're easy to keep track of. All right? Or they do have potential structural roles. We can make these really long homopolysaccharides that are not easily digested and can form nice brickwork that the cell can use for architectural purposes. Now let's contrast that with the heteropolysaccharides. These are going to have more than one type of sugar incorporated within the main chain. And these two can be branched or unbranched. It's dealer's choice, depends on what the intended function is. And heteropolysaccharides can be relatively simple, where it's just single chains of alternating sugars, like the one shown over there on the left, or like the one on the right, we can have more complicated patterns that may or may not repeat. And these very often have roles in structure over food and energy storage. And also, particularly the crazy, chaotic branched ones, roles in recognition. So here's something else I'd like you to mull over. Consider the primary sequence options we have for polysaccharides and compare them to the primary sequence of a generic protein. There's lots of ways to make a polysaccharide. Branches, no branches, they can be homo, they can be hetero. We don't have the same degree of flexibility with proteins. And I want you to speculate, why do you think that is? I mean, proteins are known as the doers of biology. You would think having all of this flexibility and these options would be more beneficial to have in a protein than a starch molecule. So think about that, and if there's time, we will also debate that a little bit in class. Of the polysaccharides, starch is notable because it's a homopolymer of glucose. Specifically, it's the storage molecule for alpha D glucose. And starch can have many structural isoforms, such as amylose, which is the simplest. Amylose is going to consist of long, unbranched glucose chains that are held together through what is known as alpha-1,4 glycosidic bonds. What do I mean by an alpha-1,4 glycosidic bond? Take a look at this figure on the bottom left. It's the structural diagram for amylose. And what I'll do for you is highlight C1 and C4. Remember that C1 is always going to be the anomeric carbon, the one that is between the two oxygens of the hemiacetal. And I'm going to count around the ring. And there we have C1 and C4 labeled. I'll do the same over here and over here. And what bonds do these ends participate in? I'm highlighting the glycosidic bonds that connect the C1 hemiacetal to the C4 hydroxyl of the next sugar unit over. And that's what we mean by an alpha-1,4 linkage. It's like conjoining two glucoses together to alpha glucoses, head to tail. And not dissimilar to the amide bond that constitutes the polypeptide backbone, the alpha-1,4 linkage is going to have preferred phi and psi angles. It's going to have dihedrals associated with it. And the preferred dihedral angles cause these amylose chains to adopt a helical chain geometry, which you can get a nice look at over here. It's not proper to call this an alpha helix. The alpha in alpha helix is different from the alpha and alpha-1,4 linkage, but they do have a common corkscrew-like geometry. And what prompts amylose chains to wind up in this manner is hydrogen bonding interactions between hydroxyls of adjacent monomer units. Those forces causes it to coil up. I will also mention that amylose is a large component to plant starch. Another component of plant starch is the next level up, amylopectin. Amylopectin has a lot of similarities to amylose. It's going to largely consist of glucoses held together through the alpha-1,4 linkage, but we're going to introduce some branch points. And these branches are held together with the alpha-1,6 linkage. Let's take a look at this diagram to see what I'm talking about. I'm going to highlight the alpha-1,4s in yellow. And let's do blue for the alpha-1,6. And we can count it out. 1, 2, 3, 4, 5, 6. That's where the C6 hydroxyl is, and that's going to form a glycosidic bond with the C1 hemiacetal of the branching glucose unit. You can get a visual of how that branch would continue the corkscrew geometry over here in that figure labeled B. And again, this is very abundant in plant starches, like potatoes. The last kind of starch is animal starch, aka glycogen. And this is the most branched. And it still will consist of a combination of alpha-1,4 linkages and branches in the alpha-1,6 position, but there's just more of these. Doesn't happen every glucose unit down the road, but at a much higher frequency than amylopectin. So you may be wondering, why are we fussing over the amount of branches? Well, that's a way that different organisms have tuned the way we store glucose. Let's draw some amylose fibrils up here as nice, long, elongated coils. You can sort of imagine that these are going to pack together really densely and therefore perhaps not be as soluble. Glycogen is going to form these highly dendritic structures that aren't going to pack together like brickwork as efficiently. And amylopectin is sort of the Goldilocks scenario, where the moderate amount of branching generates like an intermediate density. So at this end of the spectrum, we favor short-term storage. And high solubility. This makes sense because animals are going to be very active, and we're going to be making lots of withdrawals from our glycogen banks, or our glucose banks, excuse me. And therefore, by having all these branch points, our enzymes are able to access these glycogen particles really easily. Plants, on the other hand, are more sessile. They're not going to be as active. They don't run, jump, and do backflips. And when they make starches, their goal really is to provide enough food for, let's say, the seeds that they make so that they can be well fed as they mature into saplings. So we're favoring long-term storage by reducing solubility. The starch is packed together, and enzymes are not going to tap in and break it apart as quickly because we're going to save those for a rainy day. I think it's kind of cool that biology figured this out simply by adding in another type of glycosidic linkage. Let's now talk about cellulose, which is a homopolymer of beta D-glucose. We didn't forget about the beta version. All right, it has a completely different structure from starch. And that's because the B monomer of glucose is going to form beta 1,4 linkages. And these are going to be straight, not coiled. The linear beta glycosides are again going to recruit adjacent hydroxyl groups to hydrogen bond in a way that stabilizes the formation of fibrils. These are not flexible like a coil, but rather stiff like bricks. Therefore, they're going to pack together like so, really densely, even more dense than amylose, and therefore be very chemically robust because these glycosidic bonds are going to be even harder for enzymes to snip apart. Not impossible. There is an enzyme that can do it. It's called cellulase, but animals don't have that enzyme. So therefore, animals can't digest this polymer. There are plenty of decomposers that can, though. And that's not the only structural homopolysaccharide out there, aka fiber. Another kind, a less appetizing kind in my opinion, is chitin. This is a polymer of not glucose, but a derivative called N-acetyl-glucosamine. And again, it's going to be in that beta configuration, which really promotes stiff, robust fibril formation. Check out these N-acetyl groups that have been installed in the 2 position. What's the consequence of that chemical change? Well, the N-acetyl group is going to decrease the polarity of that polymer. And this has evolutionary significance to it. Think about the last time you got trapped outside in the rain. If you're wearing cotton clothes, I bet that got pretty waterlogged and really weighed you down. Cellulose is going to be very hydrophilic. Chitin, though, is going to be the main component of arthropod exoskeletons. So I'm talking bugs and lobsters. When they get wet, if that water clung to their structural homopolysaccharides, it's going to weigh them down and make them more vulnerable to predation. N-acetylglucosamine lets the water roll right off their shells. So yet again, simple changes, big ramifications. Another structural polymer I want you to recognize are peptidoglycans. These are heteropolysaccharides that consist of repeating N-acetylglucosamine and N-acetylneuramidic acid. And these are both going to be connected via the beta-1,4 linkage, and they will be particularly abundant in bacterial cell walls. But I want you to take note that peptidoglycans are actually a hybrid polymer consisting of polysaccharide and peptide intertwined. The carboxyl group of the N-acetylneuramidic acid group is actually a site of cross-linking. Over there, we will attach what's known as a tetrapeptide. And the tetrapeptide repeats are going to be cross-linked to a scaffold. And we can see the tetrapeptide anchors in the following cartoon, which I'll highlight again in yellow. You can imagine that this copious amount of stapling that we observe between the carbohydrate portion and the scaffold region is going to make this a very robust structure. And to make that connection, the bacterium is going to use a special enzyme called DD transpeptidase. This is one of those rare and freaky instances where biology is going to use D amino acids, and D-alanine is going to be part of the substrates where DD transpeptidase staples the tetrapeptide to the scaffold region. And this is the bond that we're trying to form, basically. And check this out. The molecular structure of penicillin sort of approximates the desired substrate. Penicillin is actually a suicide inhibitor for DD transpeptidase, an enzyme only expressed in bacteria. And therefore, it's going to dismantle the bacterial cell wall by inhibiting peptidoglycan cross-linking. That's why penicillin, once upon a time, before antibiotic resistance, it was such a potent way of killing bacteria. Animals, too, can form protein polysaccharide conjugates known as glycosaminoglycans or proteoglycans. They're more or less the same thing. And these are going to be among the chief components to our extracellular matrices. The general structure of a proteoglycan is that of a bottle brush polymer, where we have a primary polysaccharide backbone called hyaluronic acid, and this is going to consist of our bottle brush backbone. And that is visualized over here in this graphic. It's the core of this kind of gross looking, honestly, a fuzzy-looking polymer, which approximates how a bottle brush looks like. It's bristles upon bristles. And what is the chemical identity of the fuzzies, to use technical terms? That is going to be a combination of the following: glucosamine chondroitin, keratan sulfate, and heparan sulfate. These are the brush bristles. All three of these. Now, both the hyaluronic acid backbone and the bristles protruding from it, they all consist of repeating disaccharides of these two sugars, hexosamine alternating with hexuronic acid. What distinguishes the bristles from the backbone is that these bristle polymers over here contain sulfates. So these are sulfonated. Therefore, proteoglycans are one of the most negatively charged molecules invented by biology. Now, I'd like to pause briefly and mention a familiar sounding compound, a potentially familiar sounding compound called heparin. This is spelled differently from heparan, but they are chemically related, because heparin is a super sulfonated version of heparan sulfates. And you may recognize this compound as a medically relevant anticoagulant. There is a very positively charged domain on thrombin, the enzyme that participates in the clotting cascade, or at least one of them. And these heparin molecules in particular do a really good job of binding and trapping thrombin within this sticky forest. And therefore, that's going to prevent them from finalizing the coagulation cascade. And that's why they're very good anticoagulants. The next time you give blood or get blood work done, check out the vacuum tubes that the technician or phlebotomist uses. The inside is usually coated with heparin, and they'll usually say heparinized on the outside of those tubes. And that ensures that the blood that they draw from you doesn't clot into a solid plug before the technician can do any experiments on it. Now, this is a distinction that's easy to mix up. Proteoglycan or peptidoglycan is mostly carbohydrate, with a little bit of protein included to help stabilize it. Glycoproteins, on the other hand, are mostly protein, with a little bit of carbohydrate there for decoration. And biology has learned to attach these glycosyl groups through O or N linked modifications. These are going to be glycosidic bonds between the sugar and the hydroxyl of a serine, for example, or also commonly the amido group of a surface asparagine. And another difference between glycoproteins and the other carbohydrate conjugates discussed so far is that proteoglycan is very structural. Glycoproteins are often very functional. And very often, glycoproteins are going to serve as antigens, aka it's going to form a sugar code. Membrane-bound glycoproteins are the way that our immune system can determine what is self and what is non-self, and several major allergens are going to be actually glycosylation patterns as well. What's also interesting, in my opinion, are viral glycoproteins, because they actually use glycosylation groups as a strategy to evade the immune system. That's why we need a flu shot every year. In this influenza hemagglutinin protein, there are multiple glycosylation sites that are shown in green. And every season, they actually mix up the sugar patterns to confuse our immune system. This strategy of changing up the antigen every season is the foundation of certain viruses to be able to evade our immune system, or immunoevasion. Glycosides, by comparison, are the simplest type of glycoconjugate. It's usually just one sugar or a couple of sugars bound to any bioactive compound. And they're seen a lot in both poison and medicine. Digitoxin, shown over here, is an example of a cardiac glycoside, which is a compound that can mess with the heart's function. This poisonous compound is found in the foxglove flower, shown over here. They're very beautiful, but as an evolutionary deterrent for animals eating the flowers, they make this highly poisonous compound. And the plants very sneakily attach it to these sugars. And what does that do? It makes the compound more soluble. It can alter its activity, and it can also facilitate cellular uptake. The animal that eats the flower is going to be tricked into thinking that, oh, there's sugars in this meal. The cell picks up the sugars, and what's attached to the sugar? Poison. In controlled prescribed doses, digitoxin can also be an antiarrhythmic agent. Doxorubicin, also known as adriamycin, is another example of a medically relevant glycoside. This compound was invented by bacteria. Bacteria are constantly trying to kill each other, and this is a really potent DNA intercalator and topoisomerase inhibitor. It prevents competing bacteria from undergoing cell division. And medical professionals realized that we can co-opt this technology and use it as chemotherapy and prevent cancer cells from multiplying. This part of the drug is going to intercalate and bind the base pairs of the DNA, and the sugar is going to help with the uptake and help that molecule bind the phosphate backbone. This compound is also deeply red colored and has some pretty nasty side effects, so lots of medical professionals like to call it the red devil. Pretty much any class of biomolecule can be glycosylated, and lipids are no exception. We will talk more about them later in this course, but an example of a glycolipid I'd like to discuss right now is the blood type antigen, the ABO blood system. These are membrane lipids that possess intricate surface glycosylation patterns that our immune systems are constantly surveying. And check out their molecular structures down here, and you'll appreciate just how subtle the differences are between them. For example, the A and B antigens have pretty much identical primary structures, their glycosylation chains are going to both end with this galactose-fucose branch. The difference between them is, though, that the B antigen branches off with a galactose, and in the same position in the A antigen, it's the slightly different N-acetyl-galactosamine. As for the O antigen, sometimes people think that there is no antigen. That's not true. It's just that there is no branch at the ends, and this site is not occupied. Regardless, in all three cases, the differences between them amount to a couple of atoms. And our immune system is wise enough to tell the difference. And it just takes a few atoms of difference to turn a good transfusion into a bad, life-ending one. When glycolipids are really, really large, we give them a special name and call them lipopolysaccharides. And these are quite prevalent in bacteria. I want to bring your attention to lipid A. This is a very noxious bacterial antigen that features a super long polysaccharide chain. And the reason why lipid A is insidious is that when the outer membrane has a bunch of it, these O antigens can form this slimy forest, this carbohydrate shield that protects the bacterium and acts as a physical barrier between it and the immune cells trying to pop and kill it. So again, we see carbohydrates adopting the roles of recognition and evasion. So I hope now when you think of carbohydrates, you think of them as more than candy, sweets, and potatoes. Because the glycome, which is the collective of all the carbohydrates in an organism, turns out to be incredibly complex. Yes, the cell uses carbohydrates as a food source, but we've also learned that carbohydrates can function as structural support. Carbs have a role as well in self, non-self recognition, cell adhesion, pathogen virulence, and evasion. And the list, frankly, goes on and on and on. For example, the field of glycomics is very relevant to cancer biology. As a cell becomes cancerous, its glycome will change. The glycolipids on a tumor cell's surface will be tailored to help that cell evade the immune system and promote metastasis. And there are therapeutics in development that target that aspect of cancer. And because these carbohydrate chains can be so varied and so branched and so diverse in their functionalities, carbohydrates are probably the least understood biomolecule in biochemistry.